Anthrax is a disease caused by Bacillus anthracis, a bacteria which affects the skin, lungs, and gastrointestinal system. It is caused by the entrance of spores into the cells of the human body. It enters these body cells through means of receptor-mediated endocytosis. The endocytosis of the anthrax toxin isn't different from regular receptor-mediated endocytosis. The bacteria consists of three proteins: the protective antigen, lethal factor, and edema factor. The protective antigen is a ligand that binds to the cell receptors commonly called anthrax receptors 1 and 2. The protective antigen combines with either the lethal or edema factor to form the anthrax toxin. Anthrax begins to enter the cell when the protective antigen binds to either a TEM8 or CMG2 cell receptor. Of these two cell receptors, CMG2 has a much higher affinity for binding with the protective antigen than TEM8. After binding, a section of the protective antigen is severed by enzymes to allow for the binding of the lethal factor/edema factor. The lethal factor/edema factor then binds to the protective antigen on the cell receptor, forming the toxin. Then, a SRC kinase and E3 ubiquitin protein ligase bind to the tail of the cell receptor. The SRC kinase phosphorylates the cell receptor, and the E3 protein ligase promotes the ubiquitination or degradation of it. Multiple cell receptors then pack close together to prepare for cell entry. Cell receptors enter the cell in a clathrin or actin-coated pit that then combines with an early endosome. The lethal factor/edema factor is released from the protective antigen into the endosome with other lethal and edema factors from previous cell receptors. When the early endosome turns into a late endosome, the lethal and edema factors are released into the cytoplasm, causing changes in signaling pathways in the body. Lethal factors cause the reduction of MEKs, which leads to necrosis/hypoxia. MEKs are a part of the MAPK/ERK signaling pathway, and they contribute to cell adhesion. Edema factors lead to an increase in cyclic AMP, which leads to edema. When the edema factor enters the cytosol, a calcium influx occurs, which causes the buildup of cyclic AMP. The buildup of cyclic AMP inhibits immune cells, which allows for the survival of the bacteria. In response to the toxin invasion, ATP is released from infected macrophages, and it is sent to another macrophage, which turns into an inflammasome to warn other cells and increase their resistance to the toxin.
Oddly enough, despite being around for so long, not much is known about the internal signaling pathway of the anthrax toxin. Although scientists do know of one protein in the internal signaling pathway, the LRP6, which is part of the WNT signaling pathway. It binds to the toxin receptor and then becomes phosphorylated. Anthrax receptors can also regulate the amount of LRP6 in cells, which can affect the output of the WNT signaling pathway. The anthrax toxin doesn't affect any one cell signaling pathway. It can affect many different pathways. Such pathways include P13K signaling, SLK signaling, MAPK/ERK signaling, and WNT signaling. These pathways affect cell proliferation, growth suppressors, and fibrosis. Many antitoxins used as treatment for anthrax, such as ciprofloxacin, don't affect the cell signaling pathway, but instead target the toxin and try to neutralize it.